Okay. So what I want to do now in the next few minutes is go through some of the cells that mediate the innate inflammatory response and how those cells get activated. And so what we mean by the innate inflammatory response is the initial signs of inflammation that happen within a few hours after initial infection. And so most of you know what these signs are. They're signs that we've all experienced. For example, when we get a splinter, you have heat, you have swelling, redness, pain at the site of that initial inflammatory site. And so one way of thinking about this, and one way I like to introduce the sequence of events that occurs during an innate inflammatory response, is to think about what happens when you get a splinter. And so here you have the healthy skin where before you got the splinter, the barrier tissue, the epithelial cells of the skin are intact. You have resident effector cells, in this case a macrophage that's just underneath the skin that's not activated but is constitutively present and will alert the rest of the body in response to damage and infection at that skin site. And so here, this is what happens when you get the splinter. It breaks the skin integrity. Associated with that splinter may be dirt and bacteria that are associated, and those bacteria will activate these innate immune cells that are in the barrier site, these macrophages, to make molecules like cytokines and chemokines that I'll explain in a little bit more detail in a few minutes, that serve to act on local endothelial cells at blood vessels that help to make these cells leaky and attractive to other white blood cells that are circulating by, so that these white blood cells bind to the blood endothelial cells, fluid extravasates from the bloodstream into the tissue site, and you have swelling, redness at that site of tissue damage. And so all of these events happen really very quickly upon initially getting the splinter. And so let's just talk a little bit about this in more molecular detail. So here's a blown up picture of a macrophage that might be in that subcutaneous space underneath the skin. And these macrophages express on their cell surface what are referred to as pattern recognition receptors. And so there's a whole variety and different families of these pattern recognition receptors. And what these receptors do is they recognize molecules that are associated with microbial pathogens or tissue damage. And we refer to these molecules that are associated with pathogens as pathogen associated molecular patterns. What they are conserved patterns that are commonly associated with microbes. So most bacteria, most fungi, most viruses have different versions of these patterns that are recognized by these pattern recognition receptors on these innate immune cells like macrophages, and upon recognition of these pathogen-associated molecular patterns, they will activate these innate immune cells. And so some examples of pathogen-associated molecular patterns would be microbial cell wall constituents like carbohydrates and lipids, and even microbial DNA and RNA are all recognized by some of these pathogen or pattern recognition receptors. And so activation of these pattern recognition receptors is really a critical first step in activating these innate immune cells and initiating the innate inflammatory response. And not only are they important for initiating the innate inflammatory response, but they're also critical, as you'll see in later lectures, for activating the adaptive immune response. And so this is one class of these pattern recognition receptors that's been relatively well categorized, called toll-like receptors. And there's a whole variety of different toll-like receptors, abbreviated TLR, and ranging from TLR1 to TLR12 nowadays. And what these different toll-like receptors recognize, again, are these different pathogen-associated molecular patterns. So some toll-like receptors, for example, recognize cell wall constituents that are present on gram-negative bacteria. Other toll-like receptors may recognize molecules that are associated with the flagellum organelle on bacteria. Other toll-like receptors may recognize bacterial or viral DNA. And so these receptors can be present either on the plasma membrane on the outside of the innate immune cell, or they also may be present in endosomal compartments inside the cell, so that if a cell gets infected, the cell still has the ability to recognize patterns that are associated with infection of that cell. The other thing I just wanted to mention, if you think about this, is that we have inflammation that's not always associated with microbial infection. And so here is a picture of different types of inflammatory lesions. So this first one may be recognizable. This is a tonsil associated with somebody that has a streptococcal infection or strep throat. And so this is obviously associated with microbial infection. And you have expansion of the lymphoid tissue in the tonsil, and signs of inflammation is exhibited by this white exudate or pus associated with the lymphoid tissue here. And so this is kind of a classic inflammatory lesion associated with infection. But many of you have experienced other types of inflammation that's not necessarily associated with infection. So in panel B here, we have inflammation associated with a broken ankle or a sprained ankle, where the skin is intact, but yet we still have kind of a raised red inflammatory lesion. Also, there are autoimmune diseases. So, for example, in panel C, rheumatoid arthritis, it also initiates an inflammatory response that's not clearly associated with microbial infection. Panel D is a picture of an artery that has an atherosclerotic plaque lesion that also is associated with an inflammatory response, and also not typically associated with a microbial infection. Yet in all these cases, we still have activation of these innate immune cells and characteristics of an innate inflammatory response. So how does this work in the absence of microbial infection? And so the way this works is that there's, just like with microbial infection, there are pattern receptor recognition of molecules that are associated with tissue damage. And we refer to these molecules as danger or damage-associated molecular patterns, or DAMPs. And so these are molecules that are released upon tissue injury, but not typically by healthy cells or cells that are dying by programmed cell death or apoptotic cell death, but they're released by cells that are dying by necrosis or by traumatic tissue injury. And some examples of these danger or damage-associated molecular patterns would be constituents or molecules that are normally contained within the cytoplasm of cells but are released upon tissue damage. So molecules like uric acid or ATP, certain nuclear proteins or stress-induced proteins are all examples of damage-associated molecular patterns. And so regardless of whether or not you have pattern recognition receptor activation of pathogen-associated molecular patterns or damage-associated molecular patterns, they basically do two main things to these innate immune cells. So one thing that they do, in this case on the left, is activating a macrophage to internalize, in this case, a microbe and facilitate destruction of that microbe. So these pathogen recognition receptors facilitate recognition of the microbe itself and promote phagocytosis of that microbe. But the other thing that's really critical for initiating an innate inflammatory response is that these pattern recognition receptors will induce a signal transduction cascade inside the cell to activate patterns of gene transcription that allow these cells to produce proteins and secrete soluble factors like cytokines and other bioactive molecules that promote the inflammatory reaction. Okay. And so what I'm going to talk about are really three main innate immune cells that are very closely associated with acute inflammation: neutrophils, macrophages, and dendritic cells. And so macrophages and dendritic cells are constitutively present in tissues and express pattern recognition receptors, and you can think of them as sentinel cells that allow us to recognize when there may be damage to our barrier tissue and microbial infection or damage to that barrier tissue. Whereas neutrophils largely are not constitutively present in these tissue sites but are circulating in the blood, but are recruited to these tissue sites upon an inflammatory response, and then mediate that inflammatory response. And so first, just to talk a little bit more about macrophages. Macrophages are phagocytic cells. They recognize and engulf pathogens, but also release bioactive molecules, either bioactive lipid molecules or proteins like cytokines like interleukin 1, interleukin 6, tumor necrosis factor alpha are all good examples of cytokines that macrophages will produce and release into the extracellular space upon activation. Another class of molecules that macrophages can make, as well as other cells, are chemokines. What chemokines are small proteins typically that have a chemoattractant property, meaning that they're able to engage with receptors on other immune cells and recruit those immune cells towards the site where the chemokines are being produced. And so one example of a chemokine that's especially important for activating an innate inflammatory response is interleukin 8, also known as CXCL8. And so these inflammatory cytokines produce kind of a characteristic acute inflammatory response. And when these cytokines are released systemically, they can produce what's called an acute phase response, characterized by the production of what are called acute phase proteins. So one of these cytokines that we mentioned that the macrophages make upon stimulation through their pattern recognition receptors is a cytokine called interleukin 6. And what interleukin 6 can do when it's systemically circulated is engage with receptors on the surface of hepatocytes in the liver and induce the production of those by those cells of acute phase proteins like C-reactive protein or fibrinogen, or another protein that's referred to as mannose-binding lectin that helps activate the lectin pathway of the complement cascade. And these are all proteins that at one level or another have an antimicrobial function, either in terms of blood coagulation like fibrinogen, or C-reactive protein or mannose-binding lectin in terms of activating complement to a certain extent. But they're also used as biomarkers for when you have a systemic inflammatory response, meaning that they're used clinically, measured in the serum as a indicator of whether or not somebody is undergoing an inflammatory response. Okay, so during kind of an acute inflammatory response, one of the key steps involved in this response is the recruitment of neutrophils from the bloodstream into the site of peripheral inflammation. And so the first step in this is that neutrophils first have to be made in the bone marrow. They're released from the bone marrow into the blood circulation. And then if there's an inflammatory response, the macrophages have released proteins that act on the local vascular endothelium, and these vascular endothelial cells become leaky and express adhesion molecules that help the neutrophil that's circulating by recognize this is a site of active inflammation and migrate from the bloodstream into the site of peripheral inflammation. And so you get kind of many neutrophils that are circulating and get recruited into the peripheral tissue space, where they will, in the case of a bacterial infection associated maybe with a splinter, they will phagocytose those bacteria. And what happens then is that they will also release enzymes and molecules in that extracellular space that will not only kill the bacteria, but result in tissue damage, including damage of themselves, and there'll be lots of tissue debris at that site. And so this is also characteristic of an innate acute inflammatory response. And so this is just a slide to show you in a little bit more systematic way the steps that are involved with recruiting neutrophils that are circulating in the bloodstream and how those neutrophils get out of the bloodstream into the site of peripheral inflammation. And you can divide these the steps involved into this process into really 4 main steps that are mediated by different molecular interactions by molecules expressed on the surface of the neutrophil engaging with molecules that are expressed on the surface of the endothelium, especially when the endothelium is in the presence of an acute inflammatory response. And so the endothelial cells will express proteins on their cell surface that mediate this initial rolling reaction, subsequent activation of the innate immune cell, in this case a neutrophil, arrest of the neutrophil or stopping of the neutrophil at this point on the activated endothelium, and then finally, in response to chemokines that are being produced in the extracellular space, the neutrophil will diapedese or migrate from the bloodstream through the endothelial cells and into the peripheral tissue here. And so all of this process is regulated to a large extent by chemokines and other chemoattractants and adhesion molecules that are produced in response to that initial inflammatory response. So in response to pattern recognition receptor activation of macrophages or dendritic cells that are constitutively present in the tissue. So what this is just a little movie to kind of show you how motile the neutrophils and other white blood cells can be. And in this case, what you'll notice is that the neutrophil is moving in response to molecules that instead of being produced by the innate immune cells, but in this case are actually being produced by bacteria themselves. And so some bacteria produce chemoattractant molecules referred to as FMLP, and neutrophils express receptors for that molecule that induce migration of the neutrophil towards the bacteria.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. A Staphylococcus aureus bacterium has been added. The bacterium releases a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacterium. The bacterium, bounced around by thermal energy, moves in a random path, seeming to avoid its predator. Eventually, the neutrophil catches up with the bacterium and engulfs it by phagocytosis.
So that's an example of how neutrophils can move, and they're regulated to move that way by chemoattractant molecules and chemokines made by innate immune cells, but also can be made by microbes themselves. And so the next question, or the next step in this process, is, well, what happens when the neutrophil catches the microbe? And again, it's really based on the expression of pattern recognition receptors by the neutrophil that help the neutrophil, when it interacts with the microbe, physically engage with the microbe and internalize it or phagocytose it, where it can bring it into a microenvironment in the cytoplasm that is toxic to the microbe and will kill the microbe. And so this process of local inflammation is really a critical thing because it helps to contain initial infection at that site. And so that initial increase in vasodilation and vascular permeability that allow the recruitment of cells and fluid into the site of peripheral inflammation causes these signs of acute inflammation, but also facilitates the clearance and localization or containment of the infection at that site. And it prevents the induction of systemic inflammation. Because often systemic inflammation is associated with a more severe illness and sometimes with a high morbidity and mortality rate. And so there's one kind of classic syndrome that's associated with a more systemic infection that's referred to as sepsis. And so when you have a systemic bacterial infection activating all sorts of innate immune cell activation pathways and large amounts of systemic production of these bioactive molecules like tumor necrosis factor and IL-6 and other molecules, that promotes a systemic inflammatory response that in some cases will result in what's called septic shock and is associated with a very high mortality rate. And so the importance of the innate immune response then is to prevent the systemic infection and help limit the cascade of events that results in a systemic inflammatory response and ultimately potentially sepsis. So just to review the big picture of this innate inflammatory response and what we've talked about in the last few minutes is you have these barrier cells that are present. And as a consequence of disruptions in these barrier cells, you have activation of innate immune cells that are constitutively present at the barrier site. You may have local infection of these tissues. And as a consequence of this local infection of these tissues, pattern recognition receptors will activate these innate immune cells to make bioactive molecules like cytokines and chemokines. Complement may be activated, and that will facilitate the recruitment of additional innate immune cells at this site. And all of this happens relatively quickly within a few hours and serves to help contain that infection. But the other thing that happens that we haven't talked about yet that we'll talk about in subsequent lectures is the activation of these innate immune cells also initiates a cascade of events that's a little bit slower in getting initiated that helps promote activation of adaptive immune cells like T lymphocytes and B lymphocytes that further help promote clearance of the infection. Because often just activation of the innate immune cell by itself may be relatively effective for initial containment, but is often not sufficient to totally clear the infection. And so just to summarize the role of the innate immune response in the overall immune response is that you can think of the innate immune response as really the first line of defense. It's relatively rapid. It initiates local tissue inflammation, and critically, its main goal is to contain the infection, to kill the pathogens that are localized, and to prevent spread of those pathogens. And then the secondary goal of that innate immune response is to regulate subsequent adaptive immune responses.